All right, so this is the beginning of the final exam review. So the first topic we'll talk about today is topic 9, and that is covering DNA replication. So first we'll start with DNA and its structure. So just to review, DNA runs anti-parallel, and there are 4 nucleotide bases for DNA. There's A and T, which go paired together, adenine and thymine, as you can see on the top, and the other 2 are guanine and cytosine. And then along the sides are the backbone, which consists of the diester and the phosphate and the phosphate group and the sugar. So right here is the sugar, and then this right here is the phosphate group. And they run anti-parallel, which means that they run in opposite directions. So the 5' end is the end with the phosphate group, and the 3' end is the end shown where it has the OH on the sugar. And yeah, so they're double-stranded and they create a double helix, like a twisted ladder. And so the 5' end is the end, like I said, over here with the phosphate. But remember that the 3' end is where the next nucleotide is going to be added to the backbone. So the 5' end of a nucleotide, which consists of the phosphate, sugar, and amino group.
Nitrogenous base.
Yeah, nitrogenous base. These 3 consist of 1 nucleotide, and so when you're adding another one up there, you're going to be adding it to the 3' here, or on this side, you'd be adding it to the 3' end down there. It's added to the 3' end of the sugar. So yeah, like I said, the nucleotides are the building blocks of the DNA. They have the 5 carbon sugar, phosphate group, and nitrogenous base. And they pair up as such, A's and T's, G's and C's. And remember, uracil, the U pairs up with adenine. Adenine.
No, everyone knows.
Oh, speed it up. Okay. Okay. So, yeah, 2 parallel. These are the.
Yeah, if you don't know DNA structure at this point, I'm praying for you.
No, I feel like it's good. It's good for learning like lagging stranded. But yeah, yeah. So this is just in a bit more detail. Again, 3' end right here, and 5' end up there. So yeah, that's basically what I was just saying. Again, you'll need to know this for later, but A's and G's are purines, and thymines and cytosines are pyrimidines. And they're easy to tell apart, but just because neither of those 2 pair with each other. Yeah, so the base pairing occurs due to hydrogen bonding between hydrogens on nitrogenous bases. So that's how like the A's and T's and G's and C's line up and connect each other. This is kind of important just to keep together the 2 strands, but helicase will come in later for a variety of reasons like DNA replication or fixing different mutations and such, and that's what it'll be breaking. And so the bonding in the strands occurs because of the breakage of, or like this, the oxygen of the phosphate group is going to be pairing with, or yeah, I'm sorry, of the sugar is going to be pairing with this phosphate, and it'll create the phosphodiester bond between the 3' end and the 5'. And the strand that's already established, if there is one, will be like attached to this one up here, as you can see by the DNA being labeled there. So remember, it always gets added to the 3' end. Yeah, so this is just a quick recap of that. So the way that DNA replication works, obviously, how the DNA makes it through to the daughter cells is that the parent strand is separated into 2 single strands, and then there are copies made based on those as templates. So as you can see here, the light purple is the new strand. So each time the cell divides, one strand comes from the parental, one is new, and so each time they split, some of it is conserved, but only half each time. And that's the semi-conservative model. DNA polymerase proofreading. Oh, whoops. Did I skip ahead? Should I like?
This is where you are.
What's that?
Next slide. Next slide.
I don't know why proofreading comes before DNA replication.
I don't know. Just skip it.
Yeah. Okay, I'm gonna skip through that. So DNA replication. So what happens is at the end of S phase, we have the pre-IC complex binding with at the origin of replication. Once it fires, then helicase will move down on either side and unwind at each replication fork. And so every origin of replication is going to cause this bubble as shown, and on each side there'll be a replication fork with the helicases, and then on the top and bottom, depending on whether it's a lagging or leading strand, there will be DNA polymerase working in either sections or fully through. And so once a DNA origin of replication fires, it can't refire again during the same S phase, so that the DNA can't be replicated more than once. And cells will have many origins of replications because the DNA has billions of base pairs. You need many of these firing in a cell in order to get all of the genome copied in a timely fashion. And just again, every origin replication has 2 replication forks moving in opposite directions. So the ORC will fire, like I said, and so this just shows that in more detail. We have the helicase unwinding the DNA strands, and sliding clamp will clasp on both sides, but on this side right here, it's a bit simpler to look at for the leading strand. The sliding clamp will clamp on and attach DNA polymerase onto the leading strand. And of course, first you have to have an RNA primer set down on the leading strand. And then after you have the RNA primer put down, that's when DNA polymerase can start adding DNA nucleotides to that, because it needs a place to start. And then afterwards, it'll get replaced. The RNA pieces will get replaced by DNA fragments. And then on the lagging strand, you'll have these RNA primers as well, but because the replication fork is moving in the opposite direction of synthesis, you will need to have these Okazaki fragments are going to be made. So you need to have many of them made over and over again as the replication fork moves this direction, because the replication fork is moving in this direction while replication is moving in this direction down here. And you'll also need DNA ligase to connect these pieces of DNA that will be the Okazaki fragments. All right, yeah, this just shows that again in more detail. The single-stranded DNA binding protein is just a protein that protects any exposed single-stranded DNA after the helicase has unwound it, just to prevent DNA damage. Yeah, it's basically. This is just like a review of all of the proteins involved with this process. Helicase we just talked about uses ATP to pull them apart, and DNA polymerase, of course, is the one building these strands. Sliding clamp helps to clamp on DNA polymerases. ssDNA binding protein just helps the DNA after it's separated from helicase to be protected. And then primase is the enzyme that will add RNA primers. And the sliding clamp loader is an ATPase enzyme that uses energy to put the sliding clamp onto the DNA. And a new clamp is required at each primer for each DNA polymerase. And a good idea to study for the exam would honestly be to remember what proteins are enzymes and what aren't, and if they are enzymes, what do they use for power? So would they need ATP or GTP, or remembering also what are ATPases, which proteins are ATPases and which are GTPases would be good as well. So yeah, I was just talking about, so leading and lagging strand, the direction of replication is going this way. And so on each side, it's going in opposite directions, right? But depending on what is labeled 5' and 3', and on each side, you might have to figure out like which one is the leading or lagging strand. And so a good way to figure this out if you're stuck is to think about the fact that DNA is always going to be synthesized from or added to the 3' end. And so if you think about that here, and think about the fact that the DNA strands are running anti-parallel. So if you look at the top strand, for example, I know it's already labeled, but just pretend it's not. So with the lagging and leading. So if you look at the top strand, it's 3' on this side, 5' on this side. And since it's anti-parallel DNA, so this over here, if we were to draw one in here, it would look, it would be 3' on this end and 5' on this end. And since you know that DNA has to be added to the 3' end, each individual nucleotide here will have the 3' end on this side. And therefore, when you're opening this bubble on the top here, you can continuously add as the direction or the replication fork opens, because you can keep adding to the 3' end on this side for every nucleotide. Whereas on this side, you have to wait for the bubble to open up a little bit more, and then you can add some more, because you can only add on the right side, but not the left, because the right side is the 3' end. So you're gonna have to keep waiting for a little bit more of a section to open up, and then you can build another section, and so on and so forth. So that's, and then since this is the top one is lagging up here on the left, and that the leading over here 5 prime or this side over here is the leading strand. That on the bottom strand, it's going to be the opposite for each one, due to the bottom strand being anti-parallel to the top one. So in this case, it'd be, it's leading on this bottom one because it's lagging up here, lagging down here because it's leading up here. And by working through the logic that I was working through before, we're able to figure out what is the leading and lagging strands based just on being told what is the 3' end and what is the 5' end on the parental DNA strands. And I guess another quick way to remember, if you want to memorize or write some down your cheat sheet, is that if you're synthesizing from 5' to 3', and for your leading strand, and if you're synthesizing 5' to 3' in the opposite direction of the fork, you're lagging. This just kind of walks through. So this is what you when you're drawing on your piece of paper on the exam. You probably want to draw out these little Okazaki fragments to represent the lagging strand, and the continuous lines to represent leading. So the lagging strand is not synthesized continuously, like I was saying. It has to be synthesized in fragments called Okazaki fragments. And so the primer, so primers need to be added. RNA primers, as you can see in blue, are added first, and then DNA polymerase is going to come in while attached to the sliding clamp, and it'll synthesize the red, which is DNA, and it will, starting off with these RNA primers, will continue off until it reaches the previous RNA primer, then it'll unattach and go to the next one, the next RNA primer. And the reason why, again, that it can't just, it can't go in this direction and go all the way on this side is because it can only add to the 3' end of the DNA. So this is just kind of what I was walking through earlier. Yeah, this is just in word form, what I was just saying earlier, walking through the logic. And so the part 2 of this is going to be DNA replication, and going into the end replication issue. So at the end of each chromosome, there are sections called telomeres, and there are specialized sections with repeated nucleotide sequences, and they're needed due to the issue from losing a lot of nucleotides due to every time you reach the end of the lagging strand, there's nowhere for the DNA polymerase or the primase to have a place to put down some primer. And so what the cell does in order to make up for this is it has telomerase, which is an enzyme that has RNA. It has a sequence of RNA built into it. And what it does is it helps extend or lengthen these telomeres, because if not, the telomeres will just continue to shrink until the actual coding regions of the chromosomes are lost. And so the telomerase keeps the telomere from shrinking too much, and so it helps extend the template strand by using its RNA as a template for DNA polymerase to continue to add on, as you can see right here. So this is the telomerase right here and the RNA within it. And then DNA polymerase will come in and continue to add on sections here of DNA. And then once that's done, it allows for primers to be added, and then DNA polymerase can add to the other side as well. Wait, actually. Oh, sorry, no. So it adds its own, I got those confused. So it adds its own, so telomerase is right here in green, and so it will add DNA nucleotides right here. So this is RNA right here, and it will add DNA nucleotides by base pairing them to the RNA nucleotides. And that's why it's considered to have reverse transcriptase activity, because it's doing the opposite of transcription, which is not something, it's not part of DNA replication, but it's part of like protein or gene expression, where DNA is translated or transcribed into RNA by base pairing. So it's the opposite. It's taking RNA and base pairing DNA to it in order to add on to this strand right here. And then DNA polymerase will come in after having like a primer put here, and it will complementary base pair to the lagging strand. Practice problems. I took some of them out to put in a Kahoot, but this one is one that's left here. So the figure shows a strand of DNA being replicated over 3 time points, 1, 2, and 3. So remember that not all origins fire at the same time in S phase. So the question is, and this is so 1, 2, and 3, and these are the origins of replications. These little bubbles here at the top. But when you look at the end result, how many origins of replications are there in the DNA strand? Anyone want to take a shot? Yep. That's right. There is 3. 1, 2, and 3. And so the origins of replications here, in the beginning, there's only 2 shown, but as time goes on, more and more of the DNA is going to be unwound by helicase at these replication forks, and the direction of the forks are going apart and going towards the other ones, and so the other bubbles. And so as the bubbles grow, you can see there are 3 here, and then after this time point, eventually they would all connect to meet, and there'd be 2 distinct strands of DNA. And that's just another diagram from before. Okay. Oh yeah, are there any questions up until this point? No questions? All right. So the next topic we'll go into, do you want to talk about this one?
Okay.
Will be DNA replication error correction and DNA damage and repair mechanisms.
You skipped it.
Okay, sorry. You can have it. Don't worry about it. You want to say here or?
All right, so polymerase proofreading. This is really the first safeguard that the cell uses to prevent unwanted mutations in the DNA. All right, so how does DNA proofreading really work? Well, we know that DNA polymerase has a 5' to 3' polymerase activity. And proofreading goes in the opposite direction. So we'd say DNA polymerase has a 3 to 5 proofreading, or proofreading is really just excising erroneous nucleotides. So we can say 3' to 5' exonuclease activity, because that's really what's happening. And because of proofreading, we take the error rate from about 10 to 8, 10 to 9, down to, 10 to 5, 10 to 6, down to like 10 to 8, which is like 100 to 1,000 time fold increase or decrease in mutation. So that's pretty good. So essentially what happens is DNA polymerase has 2 subunits or domains. It's kind of different in different species. The proofreading domain can be a subunit or its own domain. And so we take it from the polymerase domain. And when DNA sees that there's been a mismatch, it actually shifts the DNA strand, if you can see, into the exonuclease domain. And it'll cleave out that unwanted nucleotide. And the DNA will bend back into the polymerase domain. And then DNA polymerization keeps on going on, like whatever. Hope that's clear. Any questions? All right. And so now we come into damage and repair mechanisms. The first one we're going to talk about is mismatch repair. And this fixes DNA mutations, essentially. So it has to occur immediately after DNA replication because, as we talk about, I don't know, this next slide, the mechanism. Yeah, so it has to occur immediately after DNA replication because you need to be able to tell which one's the parent strand and which one's the newly synthesized strand. Otherwise, you're going to get problems. And so how do cells recognize which is the newly synthesized strand? It does it through 2 mechanisms. If you're a prokaryote, you probably use methylated strands. So bacteria methylate their DNA to protect themselves against viruses. So in the newly synthesized strand, before it's been methylated, the prokaryote can be like, that has just been synthesized because I haven't been able to put the methyl groups on yet. In eukaryotes, we do it a little differently. We use NICs. So after DNA replication, the Okazaki fragments must be ligated to get rid of all the NICs. But the ligation step occurs after all the DNA polymerization has occurred, roughly like 20 minutes to an hour. So immediately afterwards, the eukaryote will look for the strand with all the NICs. If it sees NICs, then it knows it's the newly synthesized strands. If it doesn't see any NICs, that's probably the parent strand. So why do we need to recognize which is the parent strand and which is the newly synthesized strand? Well, essentially, mutations are different from DNA damage. Mutations are not strand specific, right? If you have a mutation, like in this case where it's an A to G binding, how do you know which one is the mutation? Well, the answer is you don't. Not unless you know which one is which strand is which. If there's no repair and we keep the mutated nucleotide and the non-mutated nucleotide, half your cells in the next generation, well, why is this mutated?
No,
Don't know.
Unchanged.
But there's going to be at least a mutation in half of the daughter cells that contain the erroneous A, right? If you excise the old template strand or the parent strand, both your strands will now have the mutation, right? The only way you can preserve a 100% correct sequence is if we excise the mutant nucleotide, which is why MMR is different from all the other repair mechanisms. We need to know which strand is which because mutations are not strand specific. We'll talk about that a little bit more later. All right. Okay, continue. How does this actually work? Once we detect which strand is the newly synthesized strand, we come in on a NIC. So this would be in a eukaryote because the NIC is already there. But perhaps in a prokaryote, they use an endonuclease that makes a little NIC that gives us access to the DNA strand. Next step, we use a helicase. So the helicase is going to come along the NIC. It's going to go all the way down, releasing the newly synthesized strand. And then there's going to be another cut around here, right? So if we cut here and here, and we unwind the entire strand, we're going to be left with a single stranded DNA segment, right? And because single strand DNA is not super resilient, we have to have the SSBPs to fill in this next gap. We need obviously DNA polymerase. And with DNA polymerase, if you write DNA polymerase in the mechanism, the next thing you should write is sliding clamp, because that's what gives it affinity. So the DNA polymerase is going to go along the gap, fill in all the nucleotides, and hopefully this time the DNA polymerase puts in the right nucleotide instead of messing up. And then at the very end, right, because we go 5' to 3, so this is the primer for the DNA polymerase in this step. We're going to build all the way down, but at this 5' end, right, we can't just, DNA polymerase cannot seal the nick that's going to result here. We need to have DNA ligase, and it's going to seal the nick. And then we're going to have a brand new piece of DNA that's free of the mismatch. Questions? All right, common types of DNA. All right, so MMR and DNA proofreading, those are really for mutations, right? We're going to start talking about DNA damage. DNA damage is a physical change to the DNA that should not be present in the DNA, right? That's how you identify DNA damage from mutation. So one type of DNA damage is going to be depurination. So what occurs is that we have our nucleotide, right, with the sugar phosphate bound to the nitrogenous base. But depurination sounds exactly like what it says. It's a we remove the purine base. So this affects adenines and guanines. When depurination occurs, hydrolysis occurs between the sugar and nitrogenous base bond. And now we're going to have a nucleotide that doesn't have a nitrogenous base. This is not normal. You will never see DNA without a base, right? So this is strand specific. When the cell sees a depurination, that is strand specific. This occurs about 5,000 times a day. All right, the next type, we're going to talk about deamination. So deamination is slightly different. We have a cytosine, which is commonly found in DNA, of course. But because we're going to remove this, you take an orgo, this amine group, and it's going to turn into uracil. You don't really need to understand what happens. You just need to know it turns from a C to a U. This occurs roughly 100 times a day. And again, right, uracil is never supposed to be in DNA. So this is strand specific again. And I really like this example because it really speaks to evolution. So, one of the common questions we get, or, scientists have been asking for millennia is like, why does DNA not contain uracil? Why does it contain thiamines instead of uracils, right? Well, it's probably because of deaminations. Because if uracils were normal in DNA and the cytosine converted into uracil, the DNA would not know it would not know that a mutation had occurred because uracil is naturally found. Well, in this case, the uracil would have been natural in the DNA, but of course we know it's not. We have thiamines, which are not. All right, hope that makes sense. All right, the last base-specific one we'll talk about are pyrimidine dimers, or sometimes we just call them thymine dimers. What happens is we just form a bond between the bases, blah, 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 blah. And obviously, as you can imagine, when you force a bond between these two nucleotides, that doesn't do well for the geometry of DNA. Right? So DNA has to rotate with each helix a certain amount. But the pyrimidine dimer is like, I don't know, just like a wedge that you shove in the DNA. It's going to be a lesion. It's going to bulge out. And this is really bad because, well, if you have a bulge, then stuff like DNA polymerase, helicase, stuff like that doesn't do super well with these dimers. And not super good for you. Yeah, there you go. Questions? Hope not. All right, how do we repair depurinations and deaminations? It's going to be something called base excision repair, or BER. I don't know if it calls it that, but I guess you can. So a deamination and a depurination, they're fixed in a very similar method. But to start, let's just start with a deamination. Your cytosine is converted into your uracil. The cell detects that the uracil is in the DNA. It's like, we need to get rid of this. How do we do that? Well, first, we remove the uracil from the nucleotide. And so if you think about what that does, we create an A basic nucleotide, or the same thing, essentially, as a depurinated nucleotide. And so this is where the two mechanisms, or two, whatever you want to call it, converge. The next step, right? So we have this nucleotide that we want to remove. We're going to use two endonucleases. One's going to cut on one end, the other is going to cut on the other end, right? And then we're going to remove that nucleotide. So we need two endonucleases because we need to make two cuts. So now we have this gap. How do we fill the gap? DNA polymerase and sliding clamp. And then at the very end, you're going to have a nick on the 5' end. So you're going to need DNA ligase to seal that. And so if we look at this is a little bit more clear, I think. So yeah, at the very top, we start with the deamination. But as we go down, this is basically a depurination. And so we use two endonucleases. We cut here, we remove the base. We have this gap, so we need to fill it with the polymerase sliding clamp ligase. Okay, how do we fix pyrimidine dimers? These are bulky and they're two nucleotides, so we can't really use base excision repair. We need to use something called nucleotide excision repair. So this is more akin to MMR. So what we're gonna do is, well, first step, we're gonna realize that there is a lesion. Because it's a distortion in the DNA, the cell can readily recognize that distortion, be like, that's not okay. What do? We go on left side of the lesion, we go on the right side of the lesion, and we're gonna use two endonucleases. We're gonna make a cut on the left, a cut on the right. All right, and then we're gonna use DNA helicase. It's gonna go from one cut, pull up to the other cut, and now if you think about it, the only thing that holds DNA together is the phosphodiester bond and the hydrogen bonding. So once you cut the phosphodiester bonds and the hydrogen bonding, there's nothing holding that segment to the rest of the DNA. It's just gonna float off and get degraded somewhere else in the cell. We're not gonna deal with that. And so like MMR, we have this gap. How are we gonna fill it? DNA polymerase, blah, blah, blah. Hope you get it. Questions? We just kind of skipped through a lot. Okay. Now we're going to go on the last types of DNA damage, and these are double-stranded breaks. So these are the most serious types of DNA damage because we literally just took the DNA and we snapped it in two. Now we have two separate strands, essentially. This can occur for, any high energy particle. If you sit in the sun too long, maybe it happens to you. Maybe it doesn't. Who knows? But we still got to repair it anyways. So there are two main ways we're going to repair this. We're either going to use non-homologous end joining or homologous directed repair. So non-homologous end joining is like when you're really hungry, but you don't have time. So you just go to Burger King and you order whatever they have and they just whack something out and you just live with it because that is basically what the cell is doing. It's like, I don't really care. I do not care. I just need to fix this double-stranded break. So what are you gonna do? Well, obviously it has to first identify the double-stranded break, obviously. But next step, we're gonna chip back a little bit just to make so you see how these have like little sticky ends really? We're gonna cut them back into blunt ends again. And this obviously causes some loss in genetic information. And then once we get these two blunt ends, we're just gonna jam them back together and ligate them. And this is how we fix a double-stranded break in most of our human cells. Pros, well, you don't need a homologous strand of DNA. Probably a little bit quicker too. Cons, because you use an exonuclease to cleave back these sticky ends into these blunt ends, you lost information. Okay, for us though, that's fine. In human cells, right, most of our DNA is kind of junky anyways. Like only about 1% create proteins, another 25 create what we call, the siRNAs, miRNAs, non-gene related RNAs. So like a good chunk of our DNA is just repetitive, it's useless, it's old genes that kind of evolve into useless ones. So, for us, we cut a little bit out, not a big deal. So we use it. For stuff like bacteria, yeast, they have a lower genetic size, right? They have less useless stuff in their DNA. So they're more likely to use HDR because they are less willing to just lose genetic information compared to us. All right, so what is HDR? HDR is the fancy version. Whatever. Basically, the details don't really matter. You don't have to explain it. All you have to do is say there's a HDR requires a homologous strand of DNA. Because that's basically what you do, right? You have a double-stranded break, right? We do the same thing. We use exonucleases and we chip the ends of the double-stranded breaks back a little bit. But instead of non-homologous end joining where we just stick the two ends back together, we're gonna grab a second pair of DNA, the homologous DNA. We're gonna take one of the strands, right? And we're gonna just start building it off with the homologous DNA. So basically kind of what happens here is called like an invasion. One of the DNA strands invades the homologous DNA. It replicates a little bit more based off the homologous DNA. So we gain back what we lost. And then at the very end, we just ligate the two. Whatever. Pros, DNA repair, but without any damage. Cons, you always need another homologous DNA strand per the name.
That was the end. But we have a Kahoot after this. Are there any questions though?
Question.
Okay, this will be the end of the recording, and then I'll pump the Kahoot. Wait, did I record this? I think I may have forgotten. That's just me and Zoom. Oh no, I did record it.
Just say I'm recording and I'm meeting for all. It's going to auto record, right?